So there we go. Welcome everybody. This is our third webinar from the Embryology and PGD Academy. And this is webinar 3, Interpretation of PGT Results with Roy. We're going to learn all about Roy in a moment. I'm sorry, we've just got a truck driving past my house. It's going to be a bit noisy for a moment. So I'll keep going. So I've introduced the Embryology and PGD Academy before, but just in case you weren't at our previous webinars, back in 1987, I was a clinical embryologist, which I did for about 5 years. And in 1992, I moved into the PGD field at the Hammersmith Hospital. And in 1996, I set up the first webinar. I've just realized I haven't got my video on. Sorry, let me just put the video on. Oh, I've got the video on. Yep, sorry. Yes, I set up my first first workshop we did in embryo biopsy in 1996, and in the same year started doing master's courses at my university. And Alpesh joined very near the beginning, and we've done a huge number of workshops. I think it's well over 50 now. And then quite a few years ago, we decided to set up the Embryology and PGD Academy. We feel very passionate about training embryologists in this field. So we set up the distance learning certificate in clinical embryology, and I just want to tell you a little bit about that. And then the plan for the next couple of hours is I'm going to give a little introduction to PGT, then hand over to Roy, who's going to talk to you about interpretation of the results, and he's going to tell you about the methods that we use now in PGT, which is very much more high-tech than when I was first doing this. I used to do fish, which is not done anymore. And then we are going to do a poll and then answer the Q&A as well. So just a little bit about the certificate. We have 8 modules. This is what we feel is really essential for embryologists to learn about the technology. And we're very happy that next week our webinar is on quality assessment and culture. And next week we will also be hopefully launching module 4 of the certificate. So you'll be able to register for that. And then very soon behind that will be the last 4 modules. We're really zipping through them now. We are going to record everything by Zoom, so it's really easy and efficient for us to do that. So we're going to talk today about PGT and interpretation of the results. So I wanted to just introduce you to the PGT module. You can take this module on its own. If you're really interested in PGT, you can just take this. You don't have to take any other modules of the certificate. And in April, you have a few days left to take this module for just £100. So you will have a certificate with the module that you've taken the module. And if you do the exam, you'll have a certificate that you've hopefully passed the exam. So in this module, I will do some introduction to genetics and inherited disease, tell you all about recessive and dominant disorders, etc. And I also give a lecture about chromosome abnormalities, about translocations and aneuploidies. Darren's going to talk about the genetics of infertility. And then I do a short introduction to PGT, which is a little bit, my talk today is abbreviated from that. And then we have Alpesh talking about polar body biopsy, cleavage stage biopsy, and blastocyst biopsy. Sorry, I realized we've missed off there. We have also the talk about the practical aspects of embryo biopsy. Then we've got genetic diagnosis for PGT by, of course, the legendary Alan Handyside. And then Roy, who you're meeting today, is going to talk about PGT-A. Sorry, I always spell Roy's surname wrong, and I spelled it wrong in this slide. Gelicke talks about non-invasive prenatal diagnosis. Sarah Hillman about prenatal diagnosis. I think it's important for embryologists to understand both of these and what's offered to patients once they leave our IVF clinic. And then Karen Sage talks about genetic counselling, and then I finish off with discussion of the ethics. So missing off there was Georgia Kakali, gives a very practical discussion about embryo biopsy as well in this module. So it's an amazing module, some of the world leaders in PGT. So this is the faculty. Here's Georgia. I have included Georgia in this slide here. And the logbook So for the first 4 modules of the certificate, there are a logbook, and the logbook is where the embryologist will write down every time they do a particular procedure. We're not checking competency. To check competency, we would really have to see you in your lab do this face-to-face. But the logbook is something that's used in many different training schemes to log that you've actually achieved a certain number of the procedures. So it's a basic record that you've done these things. So we have questions on biopsy preparation, preparing the manipulator and tubing cells, embryo biopsy, tubing cells, and the validation. So to apply for the certificate or an individual module, please just visit embryologycertificate.com. And as I said, there's a special offer in April, still a few days left. You can take 1 module for £100. At the checkout, just use the code 150OFF. So this is our social media. We had devised this webinar series to help occupy embryologists during this lockdown. We heard on the chat earlier that many people are still on lockdown at the moment, and so we wanted to give you something to do, and it's been really popular. And we've got 1 left next week, which is IVF troubleshooting and culture media, and then I'm sure we will do some more in the future, but we'll see whether we're all back to work, hopefully at some point in May. The Embryology and PGD Academy have a YouTube channel, and we also are on Facebook. And I have a Joyce Harper Work Facebook page. My other Facebook page that some of you might see is very much just for social things and not for work. And Alpesh and I are on all the other social media. There are Twitter and Instagram addresses. And then these are our websites. So if you want to find out more about the certificate or about our Embryology and PGD Academy, and if you'd like to give us any feedback on this webinar, then please contact the address below. During the next 24 hours or so, 48 hours, I will email all of you who have already emailed the Academy with the login details for next week's webinar. So please do not email asking for that. Just give me a few days. I will give you the YouTube link for this webinar and tell you the login details for next week's webinar. So I hope that you can just bear with me a few days to get that done. So that's the end of my introduction. I'm now going to talk about the a little background on PGT. If I can just find where the file's gone. Everything was open and now it all disappears. Right, let's just check where that is. Okay, I'll just get that open again. Right, is that one done? Right, that should come up hopefully. Right, just give me a second. Oh, it's the first time we've had a technical hitch. I don't know why this is not coming up. Just give me 1 sec. I'll just reload that. Our first technical hitch we've ever had. Ah, here it is. There we go. Introduction to PGT. Sorry about that short delay. So many files open. Right. So it's a great pleasure to give you a short introduction to pre-implantation genetic testing. As I said, I started working on this back in 1992 when I joined Alan Handyside at the Hammersmith Hospital. So Roy's also going to mention this, but for many decades, for a couple of decades, we called this pre-implantation genetic diagnosis. And then a few years ago, a group got together and decided to rename this. So we've had to get used to calling it now PGT. But as Roy's going to explain, we have now PGT for aneuploidy screening, PGT-A, PGT-M for Mendelian or monogenic diseases, and PGT-SR for chromosomal structural rearrangements. So I will just briefly explain a tiny bit about these, but Roy's going to go into these in more detail. So PGT-M and PGT-SR are what we would previously call PGD, and they're the selection of genetically normal or carrier embryos from couples who were at risk of a known inherited mutation. So these are couples that knowingly are carrying cystic fibrosis, beta thalassemia, et cetera, or they may be known to carry a chromosome abnormality, such as a translocation or other chromosome arrangement. So PGT-M and PGT-SR are for couples who have inherited disease that we know are going to be at high chance of transmitting this to their children. So the aim for these couples is to transfer an embryo that's free from the disease. So the majority of these patients actually are fertile, excuse me, and could get pregnant naturally, but they have many reproductive options. The majority will opt for prenatal diagnosis, and in some cases they'll be able to have non-invasive prenatal testing, but some, especially those that are infertile and need IVF anyway, but also some that are fertile, will decide that they want to go through PGT. So that's a very short introduction, but they might decide to go through PGT because they have an objection to termination of pregnancy. Obviously, if they're infertile, then this is the only option for them. But some of them have got pregnant naturally before, but have had repeated terminations of pregnancy and really felt that this was not the route they wanted to go through, the natural conception and prenatal, and they wanted to explore PGT. We have those that have had repeated miscarriages and do not feel they want to start a pregnancy naturally. And we're not going to talk about this today, but we do talk about this in the module. We can do PGT for save your siblings, but that's a long story. So PGT-A, so on the certificate module, Roy gives a whole talk about PGT-A, but this was previously PGS. So this is the selection of a euploid embryo or oocyte, so one that is chromosomally normal. And almost all these patients are infertile or subfertile. And the aim here is to try and improve the IVF delivery rate or the live birth rate. Has been controversial, and I'm not going to talk about that today. I've spent many, many years talking about it. Just to say, the initial indications for PGT-A were advanced maternal age, recurrent IVF failure, and recurrent miscarriage, where the couples had a normal carrier type, so they weren't carrying a translocation. But more recently, this has been offered for good prognosis patients, for social sexing, and I've written here, everyone. So there are a number of clinics in the US now that do PGT-A for every single patient that goes through their clinic. We have a paper just submitted where we've looked at the USA data, and up until the data we looked at, there were 3 clinics doing this for every single patient, and I'm sure there's probably a few more now. So just a little bit about biopsy. As I said, Alpesh and Georgia give a very detailed description on our certificate module on PGT about embryo biopsy. Alpesh is really one of the world leaders in this and talks about this at many international conferences. So I'm just going to skim over this today. You can obviously do polar body biopsy, cleavage stage. I did cleavage stage biopsy for many, many years. That was my first job at the Hammersmith to do embryo biopsy. And then we've got blastocyst biopsy as well. Now I've written non-invasive. I think we'll have some discussions about that when we do the poll. It would be really interesting to see if anyone's actually doing it yet. It's very new, and in my view still needs a little bit of work to make this viable. Now I just wanted to finish talking about mosaicism. So when I joined the Hammersmith team, as I said, my first job was to do embryo biopsy, but very soon after that, I learned the FISH technique, fluorescent in situ hybridization. And for many, many years, I did FISH on embryos. And we had been working with the group in Maastricht, Edith Kuhnen and Joep Gerards. back in the very early 90s, and they had developed a method of spreading the whole embryo in one go onto our glass slide. So we could look at all of the chromosomes together. And this is the first time this had ever been done. All previous techniques had looked at one cell at a time. This was a highly efficient technique, the HCl tween spreading. And so this was the first paper where we looked at all of the cells from an embryo in one go. And we had seen these high levels of mosaicism. Other people who had tried karyotyping in the late 80s, such as Maria Zenzis and Ros Angel, had said that they think human embryos have a lot of mosaicism, but they'd had very low success with karyotyping. So we were really amazed to see the high levels of mosaicism. To be honest, at the beginning, I thought I was doing something drastically wrong with my fish. I kept saying, my fish is not working, everything's crazy. But then we trusted our instincts and reported this back in 1995. And obviously now the issue of mosaicism is still very much with us. And Roy will talk about this in a little bit more detail. But just to say it's present at all stages of pre-implantation development. Probably maybe all cleavage stage embryos, or almost all of them, have a few abnormal cells. Maybe we can, if you want to know questions about the possible mechanism, we can talk about that in the Q&A. Probably up to about half the blastocysts. It depends what study you've looked at. It certainly is problematic for PGT, because with any diagnostic technique where you're biopsying something, the cells you biopsied should be representative of the rest of the embryo. And obviously with our biological issue of mosaicism here, that's not always the case. But it was Francesco Fiorentino, working in Italy, who was bold enough to say, okay, let's transfer some of these mosaic embryos. If the couple don't have any normal embryos, let's transfer them and see what happens, because so many embryos, mosaic embryos, were being discarded. Francesco was really amazing to do this, and there's been many studies since then, and again we can talk about that. But now these mosaic embryos are normally transferred depending about which chromosomes are involved with the abnormality. So just some key papers. So I was part of the ESR working group on what we call then PGS. So back in 1997, I was the co-founder of the ESR PGD consortium. The paper down the bottom, you can see here, the old black and white one. Alan Handyside and I back in 1994, and then I followed this up in 1996, wrote papers on the current status of PGD, which included the world data. And so from that, we set up the consortium in 1997 and continued to collect data on that. And the PGS working group were responsible for doing the groundwork that set up the ESR ESTEEM study, which is this paper in the top, in the middle, that was published in Human Reproduction. So this is the RCT, multi-center RCT using polar body biopsy. I'm not going to talk about the results. That's for another discussion. I just want to make sure you've seen these papers. Carmen Rubio is the main RCT looking at cleavage stage biopsy. Then of course we've got the STAR trial here, which is from multi-center doing blastocyst biopsy. So down the bottom, another key paper in 2010, we looked at the randomized controlled trials doing cleavage biopsy, and this is a position statement from the ESR PGD consortium. And a paper just out last week, hot off the press, we have written about the status of PGT in the UK and USA. In the UK, PGT counts for, the data we've looked at, it was only about 2%. That has gone up. But in the USA, it's much higher than that. It's about 30% in this study and has gone up every year. So those are some key papers. There are thousands of papers on PGT, but I just wanted you to be aware of some of those key papers. So that's all I'm going to say for now. So keep putting your questions in the Q&A. I'm going to stop sharing my screen now. And here's Roy. Roy, you can just unmute yourself now. Brilliant.
Hi, Joyce.
Wonderful.
Can you hear me?
And Roy's going to, so really pleased to introduce Roy to you. Roy and I started working together. What year was it, Roy?
More than 10 years ago.
Way more than 10 years ago.
Yeah.
Brilliant. So Roy's sharing his screen now. So I'm going to turn my things off and let Roy take over. Thank you very much, Roy.
Brilliant. Can you see my screen, Joyce?
We can just make it large.
Perfect.
Perfect. Thank you so much. It was always strange talking to yourself, Roy, but we are all listening, I assure you.
Brilliant.
It's coming up. It's coming up with both. Yes, that's it. Perfect. Perfect. Good.
Perfect. Great. Thanks for the introduction, Joyce. Just a disclaimer, I'm fully employed by iGenomics. I had the laboratory here in the UK. However, I always maintain an affiliation with Joyce's group at UCL, and I'm a technical expert for the United Kingdom Accreditation Service. I always like to start with this picture taken from Alan Handy's site, and it's been now a little bit more than 30 years after the diagnosis of the first PGD, pre-implantation genetic diagnosis of an X-linked disease, and that work was published in Nature in 1990, so almost more than 30 years now that we're in business. Pre-implantation genetic testing, as Joyce mentioned, terminology or nomenclature has changed in 2017. So again, you've got under the umbrella of PGT 3 services or 3 PGTs. The first one is PGT-M, monogenic disorders. These are Mendelian single gene disorders, and this was formerly known as PGD. Then you've got your pre-implantation genetic testing for aneuploidies, PGT-A, which was formerly known as PGS or pre-implantation genetic screening. And last but not least, we've got PGT-SR or pre-implantation genetic testing for structural rearrangements that was formerly known as PGD for structural rearrangements. A little bit confusing, but it's good to be familiar with the new terminology. Now, again, as also Joyce mentioned, what are the indications for PGT-M or the old PGD? Couples that have a family history or themselves are affected by autosomal dominant disorders like Huntington disease, autosomal recessive disorders, beta thalassemia, cystic fibrosis, X-linked disorders like Fragile X, also HLA typing in combination with a monogenic disorder is also performed by PGT-M, or HLA typing alone for Xavier's sibling that has a leukemia, a non-inherited disorder. And last but not least, mitochondrial disorders, which has a lot of controversy. And these are mutations in the mitochondrial genome, still very complex to perform, and we're not going to talk about that. Now, a little bit about the evolution of the technology. So I started in Joyce's lab many years ago, doing what we called a direct mutation detection using PCR on a single cell taken from, which is the blastomere, taken from a cleavage stage embryo. So we had one shot in making this PCR work. If the PCR didn't work, that's it. We could not deliver a diagnosis to the IVF clinic, which is where Alpes used to operate or used to head. And so the whole process had low accuracy. It was stressful. It was during a fresh embryo transfer. You didn't have much time to perform the diagnosis. Things eventually over the years got better and better. And where we are at now is, so what we do now is when, so we're taking now, at least in the United Kingdom, we're taking a trophectoderm biopsy. So no longer one cell, up to 10 cells from a trophectoderm biopsy sample, and performing whole genome amplification on that biopsy. So once you've done whole genome amplification, you've got a lot of material. So you can do your downstream processes many times. You can repeat them many times. And the latest technology is carrier mapping, which is a linkage analysis technology based on SNP arrays. So the technique now has high accuracy, low ADO amplification failure, and it is with carrier mapping, it is an off-the-shelf standard and rapid protocol that can be applied to more or less all patients. So no more waiting times in order to have a workup done for PGT-M. And naturally, the embryos are frozen, and this allows time for the scientists to do the analysis and reporting. No more stress. Right. So a little bit about carrier mapping. So carrier mapping is the latest technology used in PGT-M. It is still a linkage analysis. So it is a genome-wide linkage analysis platform using SNPs. The array has around 300,000 SNPs, and these SNPs are scattered all over the genome and spanning many genes that are involved in monogenic disorders. This technique was developed by the great Alan Handyside. It's based on Illumina's beta array SNP technology, which is a brilliant SNP array technology. And for this to work, for this carrier mapping technique to work, we analyze at one go parents, which are the male and female members of the couple, a reference, which is an affected child or affected father or mother of the affected patient of the couple, or aunt, in combination with embryos loaded onto a software called BlueFuse Multi. Now, carrier mapping is the most commonly used commercially available PGT-M test now in the market. Okay, again, carrier mapping, this is how it works. It is linked based on linkage analysis, 300,000 SNPs, quick sample to result. So you've got, this is an example of an autosomal recessive disorder, cystic fibrosis. You've got father who is a carrier of cystic fibrosis, mother who's a carrier of cystic fibrosis. Unfortunately, they had a child that was affected by cystic fibrosis. And this is the region on the chromosome for the affected gene. So the child inherited the blue chromosome and the yellow chromosome from the parents that harbor the mutation. Now, what we do in the PGT laboratory is, once we've received the embryo biopsies, we analyze those embryos and compare those embryos to the affected child to see what was inherited in those embryos. For example, in embryo 1, the embryo 1 had inherited the blue section of the chromosome from dad, and the green section of the chromosome from mom, so the unaffected sections of the chromosomes, hence the embryo is unaffected. Embryo 2 is a carrier because it got the yellow section from mom. Embryo 3 also is a carrier, it got the blue section from dad harboring the mutation. And embryo 4 is affected, same as the child, because it inherited the blue and the yellow sections of the chromosomes from the parents. So it's a very robust, straightforward technique. And this is an example, and this is taken from our live cases from at iGenomics UK, where this case was a PGT-M case for an autosomal recessive disorder on chromosome 15. I can't recall what was the disorder. However, this snapshot image is taken from the software used to analyze and report PGT-M. This software is now owned by Illumina. It was developed at Bluenome by Professor Alan Handyside. So what you see, in order to perform your monogenic disorder diagnosis, the software will color code, as shown in the previous slide, father and mother. Now, in this case, the parents had a previously affected child, and the child inherited the blue chromosome from father and the yellow or orange chromosome from mother. So these is what we call the baddies. And these are the embryos now that we've processed via carrier mapping and loaded on a software, and we're comparing now the embryos to the affected child. So in this case, embryo 1 inherited the good chromosome from dad and the bad chromosome from mom. So embryo 1 is a carrier. Embryo 2 is affected, blue and orange. Embryo 3 has failed. It didn't meet the QC threshold. Embryo 4 again is a carrier, and embryo 5 is affected. And this is a type of, so this is what we call the haploblock chart, and this is what we call a detailed haploblock chart shown here for embryo 5, in order to zoom in and check all the SNPs on the chromosomes for this embryo and make sure that there was no recombination within the gene of interest that happens here in this case. So this is where the gene is. These are regions flanking the gene, 2 megabase upstream, 2 megabase downstream. So we take all of this into consideration, and this is shown here for embryo 5. So embryo 5 has inherited a lot of SNPs from mom of the blue type, a lot of SNPs from dad of the blue type, a lot of SNPs from mom of the orange or yellow type. So we are definitely sure that embryo 5 is affected. So this is karyomapping and diagnosis of monogenic disorder. But karyomapping, being a SNP array, is a very powerful technique. Not only you can diagnose monogenic disorders, but you can also detect aneuploidies, especially meiotic aneuploidies. So this is taken from the same software, but from a different view of that software called the cytochip view. And in this what we call log R ratio, you've got all the chromosomes, the 24 chromosomes, the 22 autosomes, and the XY chromosomes, and you are supposed to get a straight line, and this shows the signal intensity of the SNPs on the array. So if you've got a straight line, it means that you don't have a gain or loss. However, in this case, you've got an elevation or an increase in signal intensity for chromosome 16. So this is in another view from that software, shows you like orange dots. We're not going to go through the details. And in the detailed haploblock chart view that was shown in the previous slide, we see that the discrepancy in the chromosome inherited from mom, showing a typical trisomy of chromosome 16. So not only that karyomapping detected the trisomy, it also we know where the trisomy originated from. So mom contributed 2 chromosomes 16 to the embryo. So it's maternal. This trisomy is maternal. Now this is another example of an aneuploid sample of a loss, of a chromosome loss. In fact, 2 chromosome losses here. And you can clearly see here a dip in chromosome 2 and a dip in chromosome 4. This is observed as a loss of heterozygosity in another view taken from the software. So you don't have heterozygous SNPs because you only have 1 chromosome. So that's why you have a loss of heterozygosity here. And the detailed haploblock chart, you can see the chromosomes inherited from the parents. In this case, you have a discrepancy in dad's share of chromosomes. So we were able to determine that dad did not contribute a chromosome 2 or a chromosome 4 to the embryo. So it's a paternal contribution of a chromosome loss, or losses in this case, because there are 2 chromosomes. Right, so now this is a sample report, so issued by iGenomics to the IVF clinic. And us being an ISO 15189 accredited laboratory, we fulfill all the criteria of a genetic diagnostic report. So you've got a table here showing the patient demographics, including the patient mutation. In this case, it's a cystic fibrosis, where parents are where one of the parent is a carrier, the other parent is a compound heterozygous, is affected. So one parent is carrier, another parent is affected. And the sample type, date of biopsy, date of receipt, report date and time are also displayed, however anonymized here or covered, and clinician, biopsy performed by whom and tubing performed by whom. So that's important. We display this information on our diagnostic report. And then the important bits is the test results table. You've got your embryos here, and now here you've got your linkage analysis and what the embryos inherited from parents. So embryo 3, for example, here inherited the at-risk maternal chromosome and the at-risk paternal chromosome. So this embryo is diagnosed as affected. This column is the diagnosis for the monogenic disorders. So it's affected by cystic fibrosis. And now we have another column because karyomapping also detects chromosomal abnormalities. This embryo, we did not see any chromosomal abnormalities for this embryo. However, the final result, it's still abnormal because it's affected by the monogenic disorder. And the recommendation of transfer is a no for this embryo. So let's go to embryo 8. Embryo 8 had the bad chromosome from mom, the good chromosome from mom. So the at-risk maternal chromosome was absent. The at-risk paternal chromosome was present, rendering this embryo as a carrier. Now, however, this embryo, if we didn't perform karyomapping, this embryo would have been transferred. However, we highly doubt that this embryo is going to implant, or if it implants, it's going to miscarry because this chromosome had the monosomy 18, and there's no way that the embryos can survive with the monosomy 18. So even though it's carrier for the monogenic disorder, the final result is abnormal, and this embryo is not recommended for transfer. So the report also shows the test description, any comments, test limitation, testing methodology, and then our signatures on page 3, which I haven't shown here. Right, so PGT-M by karyomapping, it has improved accuracy, reproducibility, waiting times as compared to conventional PCR methods, the old methods. So it is a sort of combined PGT-M and PGT-A, especially for meiotic aneuploidies that we trust. And it has extensive marker coverage, 300,000 SNPs, so consanguinity becomes less of an issue. And consanguineous couples can have their PGT-M done, and the results can be accurate using karyomapping. Now, we're done with PGT-M. Let's go to PGT-A, pre-implantation genetic testing for aneuploidies. What are the indications? As Joyce mentioned, the indications are advanced maternal age, repeated implantation failures, recurrent miscarriages, also previous aneuploid conception, severe male factor, infertility, or even patient request. We don't have that much or patient choice here in the UK. This is mostly applicable to the US and other countries. But in the UK, what we get the refers that we get are mostly advanced maternal age in combination with recurrent miscarriage or repeated implantation failures. And from time to time, a severe male factor. Okay, so in a nutshell, how does this work? So the state of the latest technology for PGT is next generation sequencing. And what happens is that we take a biopsy from a trophectoderm biopsy from a blastocyst. We amplify this biopsy. We amplify the whole genome of this biopsy. So we have loads of DNA. We perform DNA fragmentation, amplification, clonal amplification. We're not going to go through the details. We sequence those DNA fragments many times across the whole genome, and then we perform bioinformatic analysis, comparing the reads we have to a normal reference, and so the sequences are compared to the reference human genome. And nowadays, leading laboratories such as iGenomics use an automated NGS process. So our solution is that of the Thermo Fisher Scientific solution. We perform library preps, which is amplifying, fragmenting the DNA. However, the template preparation of the libraries is done on a robot, a super-duper robot that costs an arm and a leg, and sequencing is done on the latest sequencer. And Thermo Fisher has its own software for analysis, which we adopt at iGenomics. However, big laboratories such as iGenomics do not only rely on analysis softwares from the manufacturer. So we have developed, in addition to the Thermo Fisher software, our own algorithm in order to detect aneuploidies, gains and losses of chromosomes. So we have something called the Smart PGT-A, or the iGenomics a custom-made diagnostic algorithm that can reliably detect mosaicism and is used in combination with the clinical scientists looking at the results before issuing a report. So now what do you get as embryo results? So we're going to go through the different scenarios, or we're going to try to cover the different scenarios that you will get. This is what we call, this won't be included in the genetics, in the results report. However, this is what the scientist at iGenomics sees. So this is what we call an NGS profile. You've got your, it's not very obvious here, but you've got your 24 chromosomes, 22 autosomes and XY, and you've got a copy number here. So copy number 2 is what you need, right? We only have 2 copies of each chromosome. So if this trace is at the line coinciding with copy number 2, then you have no gains or losses. So this and then this embryo has two copies of the X chromosome, no copies of the Y chromosome. So this is a euploid female embryo. Now you can see here also a copy number 2 for all the autosomes, 1 copy number for the X, 1 copy number for the Y. So this is a euploid XY or male embryo. Now, this embryo here shows a single aneuploidy, so it's an aneuploid embryo, where you can clearly see for a loss in chromosome 18. So chromosome 18 has, instead of copy number 2, a copy number 1. So there's a loss of chromosome 18. So it's a 45 minus 18 XY, a male embryo that has lost chromosome 18. Complex aneuploidies are defined at iGenomics as embryos have a complex aneuploid embryos as embryos having 2 to 5 aneuploidies. So in this case, we've got an embryo that has a loss of 16 and a loss of 18. It's a complex aneuploid embryo. And then we come to E, and we're looking here at a chaotic embryo. You do get these patterns in females aged 40 and above. It's more abundant in those late advanced maternal age patients. So this is defined as having more than 5 aneuploidies, and as you can see here, there's different aneuploidies for that embryo, and definitely this embryo is not recommended for transfer. And then the technique is also able to detect segmental aneuploidies, as shown here with the loss of minus 5p. Now, if this embryo had been transferred, minus 5p is known to cause Cri-du-Chat syndrome. So a baby would have been born with a Cri-du-Chat syndrome. So again, this is the results report that you will receive, or that the IVF clinic is going to receive. Again, patient table showing patients, the patient demographics. The indication in this case was patient request. However, the patient is of an advanced maternal age, is age 38 years. The patient had a recurrent miscarriage and implantation failure. This is what we see day in and day out. We rarely see a young patient of 30 years old requesting a PGT-A. And then this is the results report. So for example, embryo 1 had a minus 16, is classified as abnormal, and it's no for a transfer. So on and so forth. This patient was fortunate, had 6 normal or euploid embryos, and then you can see here that embryo 12 was diagnosed as low mosaic aneuploid +8, and the transfer or recommendation for transfer is seek genetic counseling. So what iGenomics policy is to seek genetic counseling and consider the transfer of this embryo if there was no other normal or euploid embryos available. Okay. Again, this is in this report, you'll see the interpretation of test results for the different category of the test results, including low mosaic aneuploid and high mosaic aneuploid embryos, which we will discuss a little bit more in the following slides. We'll discuss a little bit more mosaicism in the following slides. Okay. Now, mosaicism in pre-implantation genetic embryos. So at iGenomics, being a medical testing laboratory, and after proper validations using cell lines with a known karyotype, 4 different categories for diagnosis were defined. So we have 4 different categories of mosaicism. If the aneuploidy is less than 30%, then the embryo is diagnosed as euploid. So it's a euploid embryo. If more than 70% of a certain chromosome is gained, so more than 70% of aneuploidy, then the embryo is diagnosed as an aneuploid embryo. Now this remains the 2 categories of mosaicism. The low mosaic embryo is an embryo that shows 30 to 50% aneuploidy for a certain chromosome. So this embryo could be considered for transfer in the absence of any other euploid embryos after going through genetic counseling. Now, the high mosaic embryos are embryos that show 50 to 70% aneuploidy. And the policy for iGenomics is not to transfer these aneuploid embryos. So you still get under the transfer column a no, and not seek genetic counseling. And this we can see here the different diagrams that we used in our validation of mosaicism. So what we used, we combined euploid cell lines with aneuploid cell lines. In this case, it was a cell line having a trisomy 18. So in this case, we took 10 cells, we isolated individually 10 cells from a euploid or a normal cell line. That is XY, that is of a male gender. And you can see here that the profile is good, is at the baseline with copy number 2 for the autosomes, 1 copy for the X chromosome, 1 copy for the Y chromosome. Now, when we took 5 cells from the euploid cell line and combined it with 3 to 5 cells from the aneuploid cell line, or a cell line that had a plus 18. What we saw here, forget about the sex chromosomes, what we saw here is an increase in ploidy for chromosome 18. So this is the category of low mosaic between 30% and 50% ploidy. So 3 to 5 cells from the aneuploid cell line combined with 5 to 7 cells from the euploid cell line. Now, if we combine 5 to 7 cells, if we took 5 to 7, when we took 5 to 7 cells of the aneuploid cell line, combine it with 3 to 5 cells from the euploid cell line, then we have something, then we have the high mosaic aneuploid scenario, which you can see here. So you can see here that this elevation in ploidy is between 2.3 to 2.5. You can't really see it here, but this elevation of ploidy exceeds 2.5. So it's between 2.5 and 2.7, or 50 to 70% increase in aneuploidy. And then if we took all the cells, or the 10 cells from an aneuploid cell line, you have a copy number 3 and a full trisomy 18. So this was our validation, and I'm going to show you here live cases or live examples from cases we had at iGenomics UK. And in this case, you can see an embryo which has pretty much a normal NGS profile. All the chromosomes except for chromosome 8 are on the baseline, copy number 2. But you can see here that chromosome 8 has a dip in the number of reads, or in the ploidy. So this we characterized, or we diagnosed, as low mosaic aneuploid -8. Again, this embryo, in the absence of euploid embryos and after genetic counseling, could be considered for transfer. Okay. Now, this is another example where all the chromosomes had a copy number 2. However, chromosome 22 had an increase of around 2.6 or 2.7. So this embryo was diagnosed as a high mosaic aneuploid embryo. And the recommendation for this embryo was no, for transfer. So no transfer for this embryo. Now it's not so bad. We thought that mosaicism in pre-implantation embryos from a blastocyst or taking a TE biopsy from blastocyst would be the end of PGT-A, but it's not so bad. So at iGenomics, we looked at between October and December 2018, we looked at more than 30,000 biopsies, and the incidence of mosaicism was 5.8% for both low mosaic and high mosaic embryos. So for the controversial result of a low mosaic embryo, this was 3.8%. So it's not so bad. It is not the end of PGT-A. And this was done across 9 different diagnostic laboratories of iGenomics, and it was highly consistent. And that's it for me. Thank you, and I guess Joyce will take the helm now.
Hi, yeah, thanks very much, Roy. That was wonderful. We'll just stop sharing, and I think you have to press stop sharing, and then we'll get yourself, me, and Alpesh's videos back on. It's amazing how complicated all of this has got since the early days of looking at a few dots on fish.
Yeah.
It's so high tech. So, yeah, brilliant, Roy. We're just going to do the poll now to get some interaction. I always say I don't like just lecturing to students. We want to hear from them and have some interaction. So I'm going to launch the poll. So all the people on board now, we've got 2 questions. How do you do your biopsy? Do you do polar body? So you might do a mix of both or several, but put what you do most of the time. What's most of your embryo biopsies? So, right, we're going to start getting some answers now. So there's polar body cleavage, blastocyst drilling hole on day 3, blastocyst drilling hole on day 5, and non-invasive. And the second question is, what is your clinical practice for mosaic embryos? To transfer, to vitrify, to discard, or other? So we'll have some discussions about what the other are. So remember, add any questions you've got to the Q&A. I found one in the chat, but I'll bring that forward. There's about another 40 people still to vote. Last ones, have we got some? So at the moment, let's just have a look where we are. So only 1 clinic's doing mainly polar body biopsy. There's 8%, 6 clinics doing cleavage stage, or 7, another one there. About a quarter, 22% are doing blastocyst biopsy drilling a hole on day 3. So vast majority are doing blastocyst biopsy drilling the hole on day 5. There are a couple doing non-invasive and a couple doing others. So I'd love to hear from you. If you want to type in, if you're doing non-invasive, please tell us more. We'd love some discussions about that and other. And then about the mosaic embryo. So obviously, Roy talked a lot about this. So only 14% said they transfer, 36% say they vitrify, 18% discard, 32% other. So we'd love to find out what the other are. So I'm going to end the poll now. I think I can share that with you. So you'll see just majority of people doing blastocyst biopsy on day 5. But I'd like to hear from those doing non-invasive and those doing other. And the majority of people with mosaic embryos are actually vitrifying, with about equal amounts transferring and discarding. So please remember, if you put your questions, put them on the Q&A, not on the chat, so we can manage them quite easily. So Alpesh, did you want to say anything about the biopsy? So do you want to quickly say how you do biopsy? We've got 62% doing biopsy drilling the hole on day 5. Oh, we just unmute you, Alpesh.
Yeah, can you hear me?
Yep, perfect.
I personally prefer making the opening on day 3. That's simply how I've learned it, and that's how I've continued to carry out the procedure. But I equally make openings on day 5 for patients who've got embryos frozen, of course. So say, for example, in a cycle, a patient has 7-8 blastocysts and they freeze them, they don't get pregnant or have a miscarriage and decide to have PGT-A, which means that we have to defrost these embryos anyway, and then make the opening on day 5. So I'm comfortable with either. As a matter of choice, if I was given in a fresh cycle what to do, I would prefer to make the opening on day 3. For me, I feel that the biopsy procedure is much easier if I make the opening on day 3, and the trophectodermal cells are much more loosely elastic rather than too cohesive when you don't make the opening on day 3.
Brilliant. Okay, we can see if there's any questions as we go through. So Roy and Alpesh, so the clinical practice for mosaic embryos, only 14% transfer. Obviously, it does depend on what the mosaicism is, but 14% say transfer, 36% vitrify, 18% discard, and 32% other. Would you like to make any comments about that?
Roy, do you want to go first?
Yeah, Roy, go
Yeah. So I'm interested in knowing of those who opt for transfer, which category of mosaicism do they transfer? Is it the low or is it the high? This is what's interesting for me. And what's other? So yeah, what do you think, Alpesh?
So I think from a clinical application perspective, I mean, we're a very new clinic, but we haven't been in a position whereby we've had to make a decision on mosaic embryos to transfer or not. Certainly, as a rule of thumb, any mosaic embryos doesn't get discarded. We would first advise the patients to have genetic counseling, which iGenomics has a good support system. We use iGenomics, and they have a good support system to recommend a genetic counselor. So that would be the first port of call, the couple having genetics counseling. As a clinical policy, I don't believe we would be transferring any embryo whereby the mosaicism is to do with the viable trisomies. So that would be our kind of take on it, if we were ever to be in that position.
Okay, so Anna has said, we transfer mosaic embryos depending on the chromosomes involved after genetic counseling, and if the mosaicism is lower than 50%.
Yeah, the low mosaic, yes.
Yeah. So I'm just
that's sensible.
Checking on the chat, but just remind everyone to put questions on the Q&A. There was a question about why, Alpesh, why day 3 and not day 4 for the zona opening?
You can do, absolutely. Whether you do it day 3, whether you do it day 4, it doesn't matter. Ideally, the whole point at making the opening at either cleavage stage or day 4 is that you still have a perivitelline space there. So you can easily apply the laser without worrying about damaging any cells as a result of laser application. So it doesn't matter. I've also made openings on day 4 as well. You know, if it's a Sunday and I don't want to go into the lab just to do a hatch on the embryo, I will make it on day 4 on Monday.
And Rashi said, does mosaicism autocorrect itself? Now, I'll start. There's been many discussions over the last 20 years about the mechanism of mosaicism and what happens to it. I've always said that I think maybe we're all a little bit mosaic. And I think having these few abnormal cells is just one of those weird things with mitosis. Whether it autocorrects itself, I mean, over the years, there were papers that were suggesting that these cells were preferentially moved off to the trophectoderm, but then there's been a lot of discussion about that. My understanding at the moment, but I'd like to hear from Roy and Alpesh, is that I don't know if there's any autocorrection. I think if some of the cells end up in the trophectoderm, I think they're more tolerated. I think if the abnormal cells end up in the inner cell mass and then key parts of where the cells go from the inner cell mass afterwards, in the development of the 3 germ layers, I think that can affect. And don't forget, when we're adults, if we take a blood sample, we're only taking blood from, we're only doing the diagnosis on our blood. If we actually biopsied our heart and our brain and our liver and everything, I think we'd see a bit of difference. And with Gudrun Moore about 20 years ago, we did a very difficult study where we looked at products that were from a termination of pregnancy. And the person doing the lab part was dissecting all the different parts of the fetuses and checking them by fish. And we found huge levels of mosaicism. And these were normal pregnancies. So I'd always hoped that someone could do this in cadavers and take samples of different parts of the germline and see ideally by karyotyping or some of the new technology, how we could do this. But what about Roy?
Yeah, so I agree with you. I don't believe there's a mechanism for autocorrection. It's just by chance. There's different types of mosaicism in a pre-implantation embryo. So you might have a mosaicism in the inner cell mass, which you will never detect because you're biopsying a trophectoderm, or a mosaicism in the trophectoderm and the inner cell mass is euploid. Then if you take a biopsy and this biopsy happens to be in an aneuploid cell line of this trophectoderm, then you have a false positive because the inner mass itself is euploid. And you've got another category which is the total mosaic, meaning that all the cells of the embryo, including the inner mass and the trophectoderm, are mosaic. So if you take a trophectoderm biopsy, it might show mosaic aneuploid, and the inner cell mass is in fact also a mosaic aneuploid. And then after that, it's a competition of cell lines who will survive more. But we believe that that the aneuploid cell line will fare less well than the euploid cell line, but it depends on the numbers. And as I've shown, mosaicism, we thought it's going to be almost 40 or 50% from a blastocyst. And what we see in the clinic is 3.8%. So Alpesh, as Alpesh said, he did not encounter a situation where he had only one embryo with a low mosaic result. You most of the time get another euploid embryo that you will transfer first. And then after that, if you run out of euploid embryos, then if it's low mosaic aneuploid, I would recommend transferring after going for genetic counseling. So I don't think that there's an autocorrection method. It's just, where the mosaicism is and the extent of the mosaicism. And we are all walking, talking mosaics. We are all walking, talking mosaics because we are made of trillions of cells, more than a trillion cells, and these cells replicate. And every time we replicate, our genome replicates, our 3 billion bases replicate, they're bound to be an error. There's loads of errors. We've got enzymes that have autocorrection. They will correct most of these errors, but some of these errors will slip. We're talking about single point mutations, but it's also similar to chromosomal breakage in mitosis. So we are all walking, talking mosaics, and the more we age, for example, Turner XO, it is known that a female, as the female ages more and more, if you take bloods, peripheral T lymphocytes from the blood, you will detect more and more XOs in the blood. So mosaicism increases with age. So mosaicism is the natural process.
I agree totally. Alpesh, what do you think?
So I think from an embryologist perspective, yes, I mean, we all hear about confined placental mosaicism in pregnancies and so on. So mosaicism, as you all agree, it seems to be a developmental process of implantation and mitosis, etc. What I'm quite interested in knowing, and Roy, you're right, obviously the practical impact of mosaicism is very rare. So clinics are hardly in that situation that they have to make that call. And usually they would either have euploid embryos to prioritize over mosaic embryos. But I think what I'm quite keen to understand, because we know that mosaicism can happen between trophectoderm and the inner cell mass, is will things change once non-invasive PGT-A comes in, because at that point you're not taking only trophectodermal DNA, you potentially also got DNA from inner cell mass. Will that show us something new that we essentially haven't seen? And the question is for Roy, that how would you be able to validate that, because, and as to what is the accurate result? Because if you're only going to see concordance with the trophectodermal cells and you're seeing something very different in the culture media, which may essentially be the ICM is doing and potentially more accurate.
Time will tell. We don't know yet, but time will tell. So can I just remind everyone, I am looking in the chat at the moment because a few people have put questions there, but please, I'm going to leave the chat in a minute and concentrate on the Q&A. So don't put any more questions in the chat. I'm just getting through these. One person said they couldn't hear, but I think everything is fine. If you are still having problems, this will be on YouTube later on, but it will be something with your computer rather than anything that we're doing. Someone said we can also have a euploid trophectoderm and a mosaic inner cell mass, which will be a false negative result for sure. We totally agree. Right, let me just check these last questions from the chat, then we're going to move into the Q&A. I am doing optimization for non-invasive, keeping up soon for publication. So we'll look forward to hearing from that. I'm sure we're going to see a lot of publications about non-invasive. Rashi said, can you please kindly explain once again how mosaic embryos are tested? Is it possible to miss the presence of mosaicism depending on the cells that are biopsied?
Yes, I guess this question is directed to me. Yes, we can miss the actual chromosome complement of the embryo. If we take a biopsy from the trophectoderm shows euploidy. However, the inner cell mass could be aneuploid or mosaic aneuploid. Then you will end up with the birth of an aneuploid baby. That is possible. This is the limitations. This is a limitation of the technology.
Or the biology of the biology. Yeah, it's not. I mean, the technology is great. I mean, it's really moved on and is so much more accurate than when we did fish, which was really a terrible procedure.
Yeah, I remember.
But this is a biology. We're stuck with biology. We can't be sure that the cells we take out are going to be the same as the rest of the embryo, certainly the inner cell mass. So
of course.
We can, as Alpesh said, let's see what non-invasive brings.
Yes. So going back to Alpesh's question or comment before, with non-invasive, we've published at iGenomics, and we've developed a non-invasive platform that we will launch after this COVID madness ends. And when you take a sample from the culture media, this sample will contain DNA that has been released from the inner cell mass mostly, and not the trophectoderm. So, and depending on the validation that the clinic will do and the concordance between the trophectoderm biopsy and the inner cell mass, we anticipate that the non-invasive is gonna be, the word is popular, but is gonna be used extensively as compared to invasive PGT-A, but only time will tell. So my answer to Alpesh's question is that in a spent culture medium or media, you will get a more representative picture of the inner cell mass from the embryo. Okay.
There's a lot of discussion at the moment of various people developing lab on a chip with the view that you take the culture media and do proteomics, metabolomics, genomics, everything. So I think it's exciting, but I think we don't know what any of them quite mean yet. So it'll be a few more years.
Of course, we just need to wait. Yeah,
we need to wait. What is the benefit test in case male is Klinefelter? Can I start? So years ago, when people looked at Klinefelter, the Belgium group in the 90s did a lot of Klinefelter patients, and they found that PGT was not of benefit. They found that the majority of embryos were normal, but that was a long time ago. I haven't kept up with where we are now. Any thoughts?
So I mean, Roy would probably know more, but again, I think on the practical application of it, you're right, Joyce, that where I was used to working before, we had done about 4 or 5 patients for non-mosaic Klinefelters. And yeah, absolutely healthy babies, unaffected. So whether it crosses that, or whether it affects the germline is the big question. But all these patients had healthy babies, even when they were non-mosaic.
Yeah, excellent.
Yeah.
Okay. Roy, yeah?
Yeah, well, I mean, I would always revert back to the Bible of, the Gardner book. And Klinefelter, during gametogenesis, the Klinefelter's chromosome will have a special type of segregation for the chromosomes, for the sex chromosomes. And theoretically speaking, you might get, euploid embryos or normal gametes, but also there are other scenarios that you might get an aneuploid gamete. So in theory, you can get both, either aneuploid or euploid. But I guess from practice, it's mostly euploid, but I don't have any, I don't have, I'm not aware of any publications checking the stats on that. But in theory, you can get either or. Yeah.
Right, we've got 29 questions in the Q&A, so we're going to try and whiz through these in the next sort of half an hour. So some of them might be repeated, but let's start. What is the validity to transfer mosaic embryos after PGT-A, and which kind, autosomal or sex chromosome? I'll leave that to Roy.
The validity, sex chromosome. All right. So then again, we do not report mosaicism for chromosomes and for chromosomes 13, 18, 21, X and Y. So we do consider sex chromosomes as, once we check that there's any ploidy changes, we report them as aneuploid. So the clinic will never transfer them. That's for the sex chromosomes. Autosomes 13, 18, 21, you will never see a mosaic result on the report, even though it is mosaic, you will always get an aneuploid result on the report. And then I would suggest the person who asked this question to refer to the Grati publication for transferring mosaic embryos. So it's a beautiful publication, beautiful work by the group of Grati et al., which classifies which mosaic embryos should be transferred and why. So yeah, I would ask the. Sorry.
I just want to say, so Roy, you reported that your diagnosis of mosaicism was very much lower than what I'd said. So I said 50% of blastocysts, but then we in the original dates had a very different way of defining mosaicism. So if we saw any results from any cell, so when we were doing the fish, even if we saw 1-2 cells that were abnormal, we said it was mosaic. So with your definition from what you've just said, anything if it's 18, 13, 21, X and Y, then you'd say they were aneuploid. So your level of mosaicism, if we were doing research, would be different to diagnostics. And also you have a cutoff. So between normal and or not saying the mosaic. And so I think that's one reason why it's a little bit different.
No, those results include 13, 18, 21, X and Y. So the 3.8% mosaicism, low mosaicism included the 13, 18, 21, X and Y. The only thing is that on the report, we won't, on a diagnostic report, we won't report mosaicism for those chromosomes, but mosaicism of these chromosomes were taken into consideration. But then again, Joyce, is the best thing is to, take a blastocyst.
Sorry, carry on. Yeah.
It's okay. Is to split the blastocyst in all of its constituent cells and do NGS for each of these cells.
Yeah. So that was the second thing I was going to say was that when I say 50% of the blastocysts are mosaic, that's looking at every single. That's research looking at every single cell. So for PGT, you're only looking at 5 to 10 cells.
5 to 10 cells. Yeah.
So that's, you're going to see lower mosaic. So if you look at all of the cells, you're going to see that.
Of course. No, yes.
So the next question is about segmental abnormality. So this has been very controversial over the years. Some people reporting it many years ago, others saying it was an artifact of their method. So the question is, do you suggest to transfer segmental mosaic embryos or chromosome mosaic?
Yes, segmental. So there are publications, there are papers saying that, where people have transferred segmental mosaics and ended up in, normal live birth. This is the least problematic category, in my opinion. So if you've got an embryo with, that is complex mosaic, so has mosaicism in 2 or more chromosomes, that's the worst embryo to transfer in terms of mosaicism. But then the next better category is an embryo that has a full chromosome that is mosaic, 1 chromosome that is mosaic. And then the best category is a segmental mosaic result. So you can transfer a segmental. So I would transfer a segmental mosaic result as compared to a complex aneuploid result or a whole chromosome mosaic result. And indeed, embryos are born and children are born normal for segmental mosaic results.
Okay, we've got a question out for a fresher. What are the tips they can take for carrying PGT in the lab? Now, I'm not sure if they're an embryology lab, then obviously learning the biopsy, but if they're a lab trying to do the diagnosis. So even when I started in this field in the 90s, I said to people, please, the embryologists should not be learning how to do PCR on fish. And now even more so. So, I mean, my answer to this would be, if they're talking about the genetic tests, they shouldn't, unless they're a genetics center, they shouldn't even remotely start to try and do this. It's, as you saw from Roy's talk, this is all very high tech. There are many labs, such as iGenomics, that will do the diagnosis for you. So I advise everyone now to work with an established, accredited lab and not try and do any of the diagnosis themselves. Roy, did you want to add anything?
No, I agree with you, Joyce.
So the biopsy's fine. Alpesh, maybe you'd like to mention now that, so we've normally been doing 2 biopsy workshops a year in the UK. We had actually not planned one for this Easter. We normally do one around Easter. And actually, I'm glad we didn't. Maybe we were predicting we wouldn't be in the lab in Easter. It would have been cancelled. But Alpesh, you might want to say something about what we're planning.
Sure, we are planning a, well, initially the reason why we didn't plan a workshop in Easter was because Joyce and I felt that non-invasive PGTA was absolutely around the corner, and teaching embryologists how to biopsy would be sending the wrong message. But I think we still are, we've got some time before non-invasive PGTA comes in. And I have been approached by quite a lot of embryologists asking if we are running workshops on biopsy. So yes, we want to run a biopsy workshop this year. We are going to be guided by how the situation is between September and November. I would have hoped that we could do a workshop in September, but again, if the situation, all the social distancing measures and everything prevent us from, then we will have to delay it. But we haven't finalized the date yet. We are going to have a workshop this year. It may be between September and November, but we have all the email addresses of all the attendees for the webinar. So whoever is keen to attend a workshop, please send Joyce an email, and then we will have a separate folder for all of those who want to be emailed about the workshop dates.
Okay, don't send me an email. Don't send me an email. My plan is in September or before. As soon as we've set the date, I will email you all. Okay. Literally, our Academy email, we're getting hundreds of emails every week. In the next 48 hours, I've got to send about 300 emails out. So sorry, Alpesh, but don't email me if you want to come to the workshop. I will let everyone who's registered for this webinar know about any workshops. Okay, so as soon as we know that it's really reliant on the COVID situation. We're ready. We've just got to wait for that. Okay, a couple of really controversial questions. In a lab, there's no PGT, but a couple come asking for a male baby. Is there any way to do the process with limited surprise, like highly active motile sperm can give a male baby? What are your opinions? So my opinion is no. Even the fish technique, Alpesh and I've done some workshops in other countries recently. A lot of people are doing fish. My view is that well, number one, I'm not a believer in doing family balancing or social sexing, whatever you want to call it. It's against my own ethical views. But also, if you are going to do it, then I don't think you should dabble. I think you should, if you're going to take the cell out or the cells out, then send it to Roy's lab and do the chromosomes. Don't just do the sex chromosomes and determine the sex. That would really grate against my own personal views. It's like doing an amniocentesis and just measuring one thing and not everything. You know, please, if you're going to do this, do the biopsy and send the samples to Roy. Any comments, Roy or Alpesh?
I agree with your choice.
Okay, right. Okay. Then there's a question about what are the reasons for giving no signal in fish? There were many reasons for having no signal in fish. It's a really poor technique. Right at the beginning, 94, I started doing fish, and I was very aware that there were a lot of false negatives and false positives. We wrote a lot about this in the 90s. There's a consortium paper that we wrote about fish, all about fish. You can read that one. That will tell you everything that you want to know about fish. But as I say, I don't think you should do it. It's a really poor technique. It gives you so little data, incorrect data. If you're going to biopsy an embryo, send it to iGenomics and get everything done. Okay, don't mess with fish. Now this one's for Roy. Can you explain non-informative ADI or no DNA detected?
Yes, of course. Right. So we'll start with non-informative. Non-informative, we do get between 1 to 2% of results that are non-informative, and these are results where the embryo, where the analysis did not meet the QC parameters. So you've got QC metrics, number of reads, signal intensity. It's just the embryo, it's just that sample had probably degradation. The DNA was degraded and we didn't get an actual trustworthy result. So we would diagnose this embryo as non-informative with a chance of rebiopsy. No DNA detected. One reason for that is when tubing the embryo, the tubed cells, got stuck into the capillary and, didn't go into the tube that is sent to a genetics laboratory. Or the DNA taken has been totally degraded. So this might lead to a no DNA detected result. Also, we would recommend a rebiopsy if possible for that embryo. What was the last category? Non-informative, no DNA detected, and...
So I've just moved the question up. Non-informative, ADI, no DNA detected.
I'm not sure what ADI is.
I don't know what ADI, I know.
Maybe allele dropout? Is that what?
Yeah, I don't know. Yeah, maybe. Yeah.
Yeah. Well, I mean, for PGT-A, there's no allele dropouts. This is for PGT-M when one of the alleles, because you're starting from a very minute DNA quantity, one of the alleles does not amplify. So ADO used to be a problem when you used to take biopsies, a single cell biopsy from a cleavage state embryo, but now we're taking up an average of 7 cells. So you're highly unlikely to get an ADO in all of these 7 cells. So when you're doing PGT-M, you will still get a result. But if you get an ADO result, then again, the only chance is to rebiopsy that embryo.
Feeling very nostalgic then. I remember in the early 90s at the Hammersmith with Pierre Ray and Pierre coming in and saying, loads of these results don't make sense. They were all supposed to be a heterozygous control, and half of them have only got normal and half of them are coming out as affected. I remember that. Now, I also remember the day I went to Joy Del Hunting said, I can't do fish. All the results are different. These are interesting memories. Thank you to Anna, who gave us a big message in capitals. You are great. Right, the next question is, Is there any way to prevent or minimize mosaic embryos? Mosaicism is a biological issue. Santiago Monet has suggested over the years that some clinics have more mosaicism than others. He suggested it could be the drug regimes or something else, but as Roy says, I think we're all a bit mosaic, but maybe if some clinics are producing more than others, there may be something. I don't know what Roy and Alpesh think.
Roy, do you want to start off on that?
Well, I mean, one of the things we do at iGenomics is that we release KPIs, key performance indicators, to all IVF clinics at the end of the month. And if we see that one clinic has a lot of mosaicism, more than 5% mosaicism, we would contact the clinic and ask the clinic what are they doing in terms, have they changed their media, have they changed the practitioner, so on and so forth. But so far we haven't been in touch in any of our clinics regarding increased mosaicism all of a sudden. But we would do if we would, if that happens. Alpesh?
Yeah, I agree. Stimulation can have an impact. Of course, lab conditions can have an impact. They say even culture media can have an impact.
Yeah.
So one thing that has definitely been debated is, does high stimulation give higher rates of mosaicism compared to mild stimulation? So it'll be good to see what emerging data we have on that.
Yeah, I haven't seen the data yet, but it would be interesting. Do you have any data experience regarding cross-contamination between embryos when using the same biopsy pipette? This is a really good question, and we always talked about this so many times. I believe some labs use a new pipette per embryo, but others don't. Alpesh?
So the answer to that question is that generally I do not change pipettes in between embryos unless I feel that there is a huge amount of debris of cells that are stuck to the pipette and will not be released when I move the pipette out of the oil, etc. So the normal process is not to change the pipette for every embryo, otherwise the biopsy can take forever. And I think even if there is potential cross-contamination of one cell, I'm sure at the genetics lab level, there is a way to identify what constitutes, I don't know, is there something you would know, Roy, from the results level, if there is a contamination of one cell compared to another cluster of cells?
It depends on which process you're doing. If it's carrier mapping for PGT-M, yes, the software will detect inconsistencies and say that there's contamination there. If it's NGS or PGT-A, we won't be able to detect cross-contamination. What we might get is mosaicism, because the cell from a euploid embryo that jumps into the biopsy of an aneuploid embryo might render the result diploidy less deviated, if you know what I mean.
What I just wanted to add there is that, of course, the biopsy is one thing, but then the embryologist washes the cells as well.
Yeah, of course, many times, many washes.
If there is another cell floating around there, which
exactly.
is a that's going to potentially not carry.
So yeah, with washing and with taking an average of 7 cells, I don't see a problem in that.
Right, so recent studies showed that the method of biopsy, pulling, may cause mosaicism. What's your opinion on that? Can you distinguish between real mosaicism and the mosaicism due to technical factors? So, I mean, I've never believed this, because mosaicism is, errors during mitosis. And there might be some errors during meiosis that have led to other things, but I can't see how what you do at a very short stage of biopsy can affect mosaicism. But I'd like to hear what the others think. So I've heard this before.
I haven't heard of the technique of biopsy causing mosaicism.
It doesn't make sense to think that.
Yeah.
Yeah. Good. Okay, we've got lots of questions, so we'll whiz through. Is there any way to identify the mosaicism as due to a technical problem? So similar thing. I can't see how that would happen. The chromosomes we analyze are mainly in interphase. They're in an interphase nucleus. You do the biopsy, it takes a few seconds, and then you're tubing up the cells. So nothing can happen to that DNA. It's enclosed. It's quite stable. So I can't see that the technique would have an effect. Knowing the difference of mosaicism levels between trophectoderm and inner cell mass, how confident are we that an embryo we consider as high or even low mosaic is truly mosaic? We end up many times with no embryos to transfer and end up in other options. Should we karyomap always?
Karyomaping is not to be, you should not do, well, it's not, I'm not saying you should not do karyomaping. It's an expensive technique. If you want to do karyomaping is the best technique because it will detect contamination. It will detect meiotic versus mitotic aneuploidies, the origin of the aneuploidies, if it's maternal or paternal, but it's not cost effective for a PGTA solution. Yeah, I wouldn't do karyomaping. And again, it's PGTA by NGS, and mosaicism is not that of a problem so far. As I saw in the recent years using NGS. Yeah.
Okay, so someone's asked for more details about the non-invasive method, but I think Roy, at the moment that's a trade secret. So the method will be that we'll collect the culture media, and there'll be different ways of doing that, and then send it to Roy.
Yes, it will be all. It is all. We've published the pilot study with 80 or around 85% concordance, and we've submitted another paper with a larger number of samples for publications. Got accepted. It's going to be published in the next month or two, and after that we will release the test will be commercially available.
And what journal's the paper that's coming out in, Roy, so that we know to look for it?
Off the top of my head, I do not know.
Don't worry.
Yeah.
But
you'll know of it. I'll send it to you right away once it's out.
Do I need to do a control group for every biopsied sample, trophectodermal blastomeres per patient, or only one control group for all biopsied samples? Now we used to do this very differently, but I think it's changed now.
No need for blanks, if that's what the person. No need for blanks. No need for blanks.
Our main worry years ago was the cumulus cells, or on cleavage biopsy. The biggest worry. There's a video that I made with Cook IVF back in the 90s, and we put some videos of biopsies on there, and they were covered in cumulus cells. And then after that, I learned the fish technique with Edith, where we spread the whole embryo. And actually, the first embryos I spread, I said, so weird. I spread an 8-cell, and I've got 20 nuclei. And then I had XX and XY, and I was like, what is going on? And then I went, oh, it's all that cumulus. And then we thought, oh my goodness me, on a fish, the cumulus looks identical to a blastomere nucleus. There's no way you can tell. And we were like, oh my goodness.
Yeah.
All these years at the beginning, this is in the early 90s, we'd been doing these biopsies and leaving on these clumps of cumulus. And if you do see this Cook video I made, every embryo has got clumps of cumulus on, but we just didn't think about it. So, we're much more efficient now than we were, for sure.
We recommend ICSI for PGT as per ESHRE guidance or guidelines. So when you're performing ICSI, I guess cumulus contamination is less of an issue. Now, if you're doing PGT-M by karyomapping will detect any contamination, whether it's paternal, maternal, or external. However, when you're doing NGS, the NGS technique won't, unless the embryo is a male embryo, the contamination is not really going to be detected. But then again, in the hands of a good embryologist, denudating, doing the denudation, you're taking an average of 7 cells from the trophectoderm, and if you get 1 or 2 cumulus cells in the sample, you're still gonna reliably deliver a diagnosis. So it was an issue when we did cleavage stage biopsies, taking 1 cell. But now trophectoderm biopsies with all the experienced embryologist and the equipment in the lab, it's not an issue.
And I jump in there. Sorry. Can I just quickly ask Roy that you said that for PGT, ICSI is advised, but I thought for PGT-A, we don't mind whether we do IVF or ICSI. Has anything changed?
We do accept, no, we do accept, we follow ESHRE, right? The ESHRE guidance or guidelines is do ICSI for all PGT cycles. However, you know, we do accept IVF samples and we don't believe it's an issue. We definitely is not an issue for PGT-M because the platform will detect contamination and the software will tell you that you've got contamination there, stop doing diagnosis. And we don't believe it's an issue with PGT-A because of the reasons I've mentioned. You're taking 7 cells, you're, you know, if you get contamination by 1 or 2 cells, you're still gonna get a result. And it's really, you know, depends on the embryologist. And, but I don't see that this is a problem. However, we still recommend ICSI because ESHRE recommends ICSI.
I mean, I was doing these biopsies in '92, so ICSI had only just started. And I can remember Alan went off to Andre's lab and Palermo's lab to see how they did it. And he came back and he said, I can't remember the number, they were doing like literally hundreds a month. And I was like, they've just started. How can they be doing so many? This was literally in 92, 93 that they really jumped in. I think Alpesh has already answered this. What is the best in PGT biopsy, donor drilling on day 3 or day 5? How can I know the inner cell mass if I do day 3?
The answer is that you're not going to know where the inner cell mass pops over from. But there are ways, even if the inner cell mass does herniate out on day 5, then there are ways to biopsy those embryos. That's a whole new discussion, really. That's more about embryo biopsy. I prefer to make the opening on day 3, but I equally know a lot of embryologists who make the opening on day 5 for the added advantage that you know where the ICM is.
Right, so we're, I'm going to speed us all up a bit. We're about halfway through the questions and we've got about 20 minutes left. What's the scale of severity? Mosaic segmental gain loss, mosaic gain loss, and segmental gain loss? That's $1 million question.
So come again. So what are the 3?
Segmental gain loss, mosaic gain loss. Oh, 2 of them are the same. Mosaic, yeah.
I would transfer the mosaic gain and not loss. But then again, with mosaicism, gains and losses. If it was not a mosaic, if it was not a mosaic result, a monosomy will always lead to an implantation failure or miscarriage. You know, it's more drastic to the embryo than a trisomy. But when you're talking about mosaicism of segmentals or even whole chromosomes, the effect, whether it's a gain or loss, according to recent studies like the Grati paper, it's the same. You're going to get the same phenotype, which we don't know which phenotype it's going to be. So I would transfer a mosaic segmental, not a full segmental, because if you transfer a segmental that has a -5p, you might probably end up with a child having cri du chat, 22q11.2 DiGeorge syndrome, 1p36 mental retardation, 15q11.2 Engelmann, Prader-Willi. Those are all segmentals, and you should not transfer segmentals.
Yeah, this really shows that this is why you need an experienced genetics lab to be doing your PGT. You can't dabble in this. You need to understand genetics is complicated. So having something like a company like iGenomics, you know all of these things and you can advise the lab what to do. So this is really, really essential. Alpesh, I know what you're going to say. Day 4 biopsy.
We don't do it here. I know in Australia, they have tried doing day 4, or what they call morula biopsy. And apparently they swear by it, that their results are concordant with day 5 biopsy. The only reason why they do day 4 biopsy is to try and avoid a weekend. But I don't have any experience, and I would just say, why would you do it? But of course, you know, if you're trying to avoid weekend work and everything, it may have its merit.
Mm-hmm. There's a question here, but I think some of it got missed somewhere. It said something about low and if there is other available. I think it must have been from something earlier on, but there's too many questions in between. I have difficulty in holding the PCR tube. Could you share how you hold the PCR tube? Is there any alternative or other option like devices? So on our Academy website, we've got a video of Alpesh doing a mock-up of tubing during a biopsy workshop. And if you take the PGT module, Alpesh did a mock-up of, in his lab, a more detailed description of how he tubes the cells. So Alpesh, just in sort of 1 minute, did you want to add anything?
Yes, there are devices that are available that aids you for the tubing procedure. There are devices available from MTG, a subsidiary arm of Vitrolife. So please ask your Vitrolife rep. MTG make a gadget. I think it's tube it or tubing it. I don't know. It's a contraption where you can lock your PCR tube and literally with one hand just do the tubing.
Okay, lots of questions about biopsy and things. So what are the criteria or requirements of working place when carrying out biopsy and tubing procedures, such as, does it require a fume hood, room temperature, clean and isolated room? Do I wear gloves? So lots of questions there. Again, if you, hate to keep plugging the certificate module, but if you take the module, Alpesh and Georgia have spent a huge amount of time going through everything you need to know with all the mock-ups about biopsy, and all of those questions are answered. Alpesh, just again in 1 minute, anything you wanted to say?
Clean air, of course, everything in the lab should be aseptic conditions. Embryologists are advised to wear gloves, definitely, because any kind of external DNA contamination, even from your skin cells, etc., may pose a problem. Tubing has to be done in class 2 cabinet. Of course, we understand that micromanipulation doesn't have to be done in a class 2 cabinet, i.e. the biopsy can be done on the bench, but the tubing definitely has to be done in a class 2 cabinet with aseptic conditions.
Lots coming up all about biopsy. Did you prefer to biopsy on day 5 and transfer on day 6? And did day 6 transfer after biopsy give good results?
So we don't do that as a procedure. We don't do any fresh transfers at all. We send our cells to iGenomics, and hence we don't have that time, just literally less than 24 hours to do transfer. And we prefer, for clinical reasons as well, to do a transfer in an FET cycle rather than fresh. Okay,
after you've done the, sorry so many about biopsy, after I've done biopsy day 5 trifectodome, do I need to keep it in the incubator or leave it at room temperature?
The biopsy itself has to be put into the freezer. So once you tube the cells, those cells have to go into a -20 freezer straight away.
Yeah, and the
embryos don't go in the fridge or freezer. The embryos get vitrified after that.
Yes, so embryos vitrified and the cells go off to the genetics lab. By making whole on day 3, there is a chance of the cell to escape from the zona. And if this happens, we'll lower the blastulation rate. As far as I know, intact cell connections promote cell-to-cell junction formation. Correct me if I'm wrong. I know what you're going to say, Alpesh, but I'll let you say it.
No, so you will not lose cells if your hole is not too big. The opening that you make is about 10 to 15 microns, just like you do with assisted hatching, a single channel. You will not lose, or you shouldn't lose cells through that kind of opening. And the gap junctions and tight junctions still continue to form. That doesn't impact on the embryo. Yes, the embryo will be herniating the cells out on day 5, but that's exactly what you want. So you can biopsy those cells.
Okay, someone said, it might be a stupid question, but why don't we biopsy the inner cell mass as well if the cells are pluripotent cells? Well, the data is that we think only a few of the cells, a handful of the cells on the inner cell mass go on to make the fetus. And I think it would take years of experiments to really see if we were doing anything. And I think no one's wanted to go there. We just couldn't do this on human embryos to check that we're not doing any harm.
Especially with non-invasive around the corner. So
yeah. I know it's safe to biopsy the day 5 early blast expansion grade 1. Oh, is it safe? Or do I need to continue to culture a few more hours? How long should I wait?
Answer is that if you have grade 1 blastocyst, you would wait to day 6 before biopsy.
Okay. So what's your opinion about non-invasive PGT-A and mosaicism issue? Will we face into more mosaic embryos? Can you tell us that yet, Roy, from your experience?
Meaning, will you get mosaic results when you're doing non-invasive? That will need to wait. But yeah, we will need to wait for that.
Yeah, for sure. Can the technique of biopsy at blastocysts by itself influence the results? In what way? So I think we said that earlier. We don't see I think any of us think that any of that, as I said, it's an interphase nucleus, they're stable. Yeah. I would like to ask regarding the biopsy, do I need to change, biopsy needle. Okay, so we've said that, we've answered that one. Between a mosaic embryo, but with good morphology, e.g. 4AA, mosaic 30 to 50%, compared to a euploid, but poor morphology embryo, e.g. euploid 4cc, which would you transfer first? That's a good question.
That's a very good question. By the law of the land, I would transfer the euploid 4cc first. We have seen pregnancies from CC blastocysts. So I would always choose euploid embryos first. Again, medico-legally, you would be in a better defense system to transfer euploid embryo first. And then after genetic counseling, consider the transfer of a low mosaic blastocyst.
And the higher grade mosaic will survive vitrification better? Probably, would it?
No, they would equally.
Equally, okay, thank you. I've never vitrified, so I don't know. I used to do lots of slow freezing. Would you ever take incomplete penetrance of a disease into account?
The incomplete penetrance of a disease. Well, luckily, the pre-implantation genetic testing laboratory will report what the embryo has inherited. Now, it is the decision of the clinician or the clinical geneticist working at the IVF center to make a decision about the disorder. If it's an autosomal dominant disorder with not high penetrance, an embryo will always inherit or not inherit. This will be detected and will be added to the report. But the decision to transfer this embryo is for the clinician or the clinical geneticist. Okay,
now we're just going to whiz through these last few questions. Roy, this is another question about the publication. Which mosaic embryo to transfer and why? Name of the publication. So have you got the PDF of the paper now that's the sort of setting the details of transferring mosaic embryos? You mentioned it earlier. Oh, you're muted at the moment. If Roy could find that Roy, if you can find that.
Yes, I've got it. Just bear with me.
You should be able to share the screen.
Yeah, just bear with me 1 second, please.
I'll just ask Alpesh. Oh no, they're the last few are all. So Alpesh, would you please inform us about the suggested blastocyst quality that we could perform PGT? Would you biopsy a 3BB embryo?
Yes, we would biopsy any embryo where we can see an inner cell mass as well. And I know I've said that we've even biopsied a CC blastocyst sometimes when we can't be 100% if there is an inner cell mass or not.
And your thoughts about rebiopsy of embryos, either at the time of biopsy, e.g. maybe due to tubing failure, or subsequently due to no DNA detected?
Yeah, we often find ourselves sometimes that we have to rebiopsy embryos when we do not get a result from a PGT-A analysis. So we may be going back to that embryo, defrosting it, and then rebiopsying it. Perfectly possible. In terms of implantation rates, yes, perfectly good implantation rates. Is there a difference in implantation rates? Potentially, because that's another biopsy on the embryo. So we don't know about that.
Did you find it, Roy? Can we share?
Trying to.
Okay, now keep
trying to. I've got it.
Oh, you got it? Let's have a go.
I'm going to share my screen and just bear with me 1 moment. Sorry.
I'll ask this one while waiting. In regard, oh, here we go. There we go. Look at that. So an evidence-based scoring system for prioritizing mosaic aneuploid embryos following PGS. So by Gratti et al. And that's in RBM Online. So that's published now. This one was one you had earlier.
2018,
yeah. Okay, thanks. You can unshare now. Thank you. Brilliant. Right, we've got a few questions left. How long can we store the biopsy in the -20 degrees before running the sample for Roy?
Right, so we did the validation. Well, if you've got a minus 20, right?
Yeah.
Right. We've done validations, and up to 2 weeks, the cells are intact, so integrity is not compromised. But from previous works and at UCL, we've kept biopsies for up to a month, and we believe that they're still, as long as you don't open the freezer often and, you know, freeze thaw, they should be okay. But the best thing is to have a -80 if you want to store it for many, many months or up to a year. But -20 up to a month with not opening the freezer often should be okay.
Brilliant. The last few questions in the last 10 minutes. Are uniparental disomies detected during PGT-M?
It depends on which technique you use. If you're using karyomapping, heterodisomy, yes, is detected using PGT-M. I've never encountered isodisomy, but heterodisomy is detected with PGT-M using karyomapping.
In case the patients have only one blastocyst from 3PN embryo and the PGT result is euploid XY, I would like to ask your suggestion. Can we transfer this embryo, or should we do more testing, or should we discard? I would transfer it, but let's see what the rest say.
Yes, I mean, triploidy 3PN can be detected by karyomapping. So we are able to detect triploidy. But if you're doing NGS, you're not going to detect a triploid unless it's a 69XXY. If it's a 69XXX, you won't know. But if it's a 69XXY and you've got a clear NGS trace, you can, by looking at the sex chromosomes, detect something that is either a triploid or a maternal contamination. But I would transfer a 3PN if this is the only embryo you've got.
What about 1PNs, Roy?
1PNs, you can detect haploidy also by karyomapping, but transferring 1PNs, I mean, I don't know, Alpish.
We did this back in the 90s. I published a paper with Robert Winston and Karen, what was the embryologist's name? Karen, anyway, we published a paper on this, honestly, in the 90s, where we did FISH on 1PNs and 3PNs, because I always thought that we were just either missing them or sometimes misinterpreting the 3PNs. And almost all of the embryos we found were normal diploid.
Mm-hmm.
I mean, that was in the 90s. So, yeah. So last few questions. You've got a young patient with high mosaicism and aneuploid. What do you think?
Well, sorry, one high mosaic result and one aneuploid result.
No, you've got a young patient with high mosaicism and aneuploidy.
What's the question?
What's the question? Mosaicism is irrelevant of age. Mosaicism happens.
So if you've got a young, if you've got any patient with high abnormalities, and if they go through a couple of cycles and they will all come out high, then obviously there's something else going on there. They're not producing normal embryos. There's definitely people that don't produce normal embryos. Yeah. And there'll be some young people, just like many young women can still have a Down syndrome child. It's, you know, one of those things. Yeah. What do you do if you don't see the cells in the PCR tube? Hope they are there or rebiopsy?
No, you don't expect to see cells every time. Of course, you aim to do the tubing under direct vision so you can see the cells come out. But sometimes the shadows in the PCR tube may not allow you to see the cells coming. Most importantly, what you need to do after releasing the cells in the tube, you need to wash the capillary a couple of times, 2-3 times, just to see if any cells were stuck in the capillary that come out. That is actually more of an important check. But no, I would not rebiopsy. Chances are 99.9% the cells are in the tube.
If I plan to rebiopsy the embryo, do I need to use the same hole, or have I to make another hole to make another trophectoderm?
Same hole.
Same hole. I know that we had all this discussion again in the 90s. How much of a concern is fragmentation when performing biopsy of borderline quality blastocysts? And can this influence the PGT-A results?
The procedure to use is if you have a lot of unincorporated cells in the blastocysts, which are sitting in between the blastocyst and the zona pellucida, you try and avoid these unincorporated cells if they come out of the embryo. Because chances are that the embryo may have expelled these cells due to aneuploidies anyway. So you want to try and make sure that they do not get taken over in the PCR tube.
Again, we looked at them in the 90s using fish, and some of these fragments definitely had DNA in, and very, very odd.
They would even be cells.
Yeah. So yeah, avoid them. Do you biopsy other embryos than 2PN? If yes, how do you justify this to patients? So we just discussed that. I think the 1PN and the 3PN. Obviously, the number one, plan A, is the 2PN. But if they've got nothing,
then 3PN, then at last 1PN.
1PN, yeah. Is it possible to send biopsy samples without the ice pack inside the box? Because the protocol of iGenomics is to send the samples inside their box with the ice pack.
Yeah, the ice pack will prevent degradation, DNA degradation. Always freeze the ice pack in your -20 or -80. And once you call for a courier, pack up, add these ice packs to your, biopsy kit, close the cooler and send it to the genetics laboratory.
Okay, last two questions, everyone. Well done. I'm sure Alpesh and Roy are exhausted. Right, this is a long one. In regarding to the non-invasive PGT, I am aware some papers did suggest to perform a laser on day 3, then further culture the embryos for 5 to 6 till day 5 to 6 for the collection of spelt culture media. However, some suggest it's not necessary to perform the laser on day 3. I was guessing the concern is to lower the risk of amplification failure. So I'm just wondering what your view is on this. If you're going to drill a hole, it's not going to be non-invasive.
Exactly. Here you go. Here you go. You've answered the
and I don't believe that the zona pellucida is a barrier to DNA coming into the.
No, it's not. No, it's not.
Right, last question. I've been suffering from a backache, so I need to get up soon. If non-invasive PGT results to be chaotic mosaic, would you perform biopsy to reassure the rest of that particular embryo? That's a good question to finish on, because just generally, will the results of the NIPGT, do you think we'd ever have to think, oh, we need to rebiopsy to reassure ourselves?
Well, it depends on what the patient wants. Does the patient want any invasive technique to be done to the embryo? If yes, I would biopsy the TE. If not, I would transfer that embryo if it was the only embryo available. It really depends. It's a decision that needs to be taken in, after talking to the patient, the clinician, and the genetics person.
Okay, well, we're almost exactly at 2 hours. Again, I don't know how we always do this, Alpesh. It's always 2 hours. We're so organized. I'd like to thank Roy very much for being our special guest today. Roy, that was really terrific. We really needed this session. A lot of really, really interesting questions. And so Alpesh and I will be back next week. This may be the last webinar for a while. It takes quite a lot of effort to get all these things together and to email everyone and to promote them. So next week, we're going to talk about, I'm going to talk a little bit about culture media. And Alpesh, was it troubleshooting or quality?
Yeah, troubleshooting and culture media.
Yeah. Okay. So we will set this up in the next couple of days, send you all an email. Don't worry about emailing me and just give me a bit of time. And then we will hopefully see some of you next week. Okay, so it's goodbye from me, and I'll let Roy and Alpesh say goodbye.
Thank you so much. Thank you.
Thank you for the invite. Thank you, Joyce. Thank you, Alpesh.
Stop recording now. Thank you very much.
Bye.